Hi everyone, my name is Amber. My project is called a miRNA-based approach to optimizing GFP expression in linearized TetR gene circuits. So it sounds like a lot, but I'll break it down in the next few slides. So first, what is the TetR system used for and what is its structure? So in the TetR system, this is a very common gene system in bacterial systems. So what we have here is a promoter that's producing TetR and another promoter that's producing YEGFP, also known as the reporter protein. So as the levels of TetR increases, it actually represses, you can see that by this red line here, it represses the YEGFP expression. So we kind of see a system of negative feedback here. And as we introduce ATC, it binds to TetR and prevents TetR from binding to YEGFP. So by doing that, increasing the levels of ATC increases the level of YEGFP. So why is this important? So we can use this system. It's very commonly used in systems biology to investigate treatments for cancer and immunoneurological disorders, because you can actually fine-tune expression of the drug based on a certain amount of inducer. And by doing that, you can determine like what is the exact threshold that this drug's effects are going to happen. So there are a lot of obstacles facing this system, however, and a major obstacle is the high stochasticity. So we can define stochasticity, or CV, as the population standard deviation over the population mean. And this is a demonstration of stochasticity on the left. So we can see when the levels of green and red protein are the same, we kind of have this uniform like yellow bacteria. But on the bottom, we have some effects of fluctuation. There's some stochasticity here. So we can see some bacteria are red and some bacteria are green. So you kind of have this heterogeneity element. On the right here, we do have a CV graph. So you can see as the ATC increases between 50 and 100, we're going to see this kind of spike in CV. And this is really not good, because we do want to minimize the amount of heterogeneity whenever we're using this gene system. Another issue with this system is, of course, the sigmoidal gene expression. So on the left here, the blue and red lines, we see this is a linear gene expression. So as ATC increases, the fluorescence is going to increase in a predictable manner. But here's what's called a sigmoidal or S-shaped distribution. You can see on the bottom, at low levels of inducer, as the ATC increases, the fluorescence does not really increase at all. And then we kind of hit this sharp dose response curve, and then it kind of flattens off again. So this is very unpredictable behavior. We don't know when it's going to hit that sharp dose response curve, so it's not really easily applicable to many real life systems. And here again, we can see this high level of variance. So my research aims to basically take this TetR gene system and optimize several elements of it to make it more applicable to usage in gene therapy. So our first aim is to introduce negative autoregulation in order to, one, linearize the dose response, which refers to turning the sigmoidal curve into a linear curve, like the one shown right here. And we also want to reduce variance and eliminate bimodality, which goes hand in hand with, of course, linearizing the dose response. And our second aim is to introduce miRNA in order to reduce basal GFP expression, or GFP expression when there is no inducer involved. And this is very good for systems that are only non-lethal at low toxins. So if we want to look at a system where, we want to look at what is the effect of this toxin on cell death, we might want to use miRNA systems. Okay, so the premise of my research is based on the idea of mass action kinetics. So this is a very common concept in chemistry. When you have two reactants, they combine to form a product at rate c. And this can actually be transferred into a system of differential equations. So d times the concentration of A, so that'll be the change in A over the change in time. This can be modeled as A, which is the rate constant, times the concentration of A times the concentration of B. And this is a second order reaction for the formation of product C. And here we also have a degradation reaction, which can be modeled similarly. This might be a system some of you are familiar with. It's the very common Michaelis-Menten system. So basically, it's enzyme forming a product with substrate, and then we take this kind of complicated system of differential equations and we put it into DISY, which is the chemical modeling software, and we can create deterministic and stochastic models. All right, so our first system is the no regulation inducible TetR gene circuit. So here we have TetR being produced and it does not repress its own promoter. So here we can see, let's take an example reaction, the promoter creating mRNA at a rate lowercase m. So the reaction can be modeled as the change in m, the big M, over the change in time is equal to the reaction rate times the amount of available promoter. And then by creating this, turning this into code, we can model huge gene systems. So this is a very powerful method of looking at large-scale gene expression. Our second model is the TetR gene circuit with negative autoregulation. So by creating this model, we were hoping to linearize the gene expression and reduce variability. So in this model, the TetR actually goes back and it binds to its own promoter and decreases its own expression. And we will see later how this works to decrease linearity. Our third model is the linearized miRNA TetR circuit. So on the left, we have the regular negative feedback one, and on the right, we introduce miRNA, which is going to bind to the TetR and YEGFP mRNA to degrade it. And this is actually blocking translation and transcription from occurring effectively. So here's our results. So on the left, we have our negative regulation system negative feedback system compared to the no regulation system. So you can see here this blue and red line that is with negative feedback involved. And this is obviously a very good, it's linear, it has an R squared of 0.99, which is very high. The L1 norm is also very, very small. So we're going to see that this is a very, very effective system at inducing linearity. Another result we observed was the reduction of noise at intermediate levels of inducers. So here we have at intermediate levels previously, again, we see this like very, very large spike. But when we do introduce miRNA, we're going to see the levels of noise decrease significantly. In fact, we do see a 7-fold reduction in CV. Finally, we tried introducing miRNA to decrease the basal expression, and we had very, very interesting results. So when the miRNA binding rate is 0, we see that the fold change is preserved, but the basal expression is quite high. And as we increase this binding rate, we're going to see the basal expression drop, which is good, but the fold change also drops, which is not as good because we want the fold change to be high in order to see kind of a difference in expression between high noise and low noise systems. But whenever the basal expression is lowered, we're also going to be able to deal with systems with high levels of toxicity. So it's kind of a trade-off between which system are you going to be working with more often. So now I ran these scans on the miRNA degradation rate. So we can see as the degradation rate increases, we're going to see a little bit of fold change recovery, as well as an increase in basal GFP expression. So really, the purpose of this, we want to kind of discover what is the best combination of binding rate and degradation rate for use in experimental kind of environments. All right, so some applications of these systems now, using the linearized gene system, this is a major breakthrough because we can finally tune and precisely study the dose-response relationship of the following major gene systems. So in development, for example, we have the muscle protein MyoD, which actually induces a bistable long-term memory response in muscle cells. So we can see with the signal Y, we have the levels of long-term memory, they increase significantly. And this kind of induces, at a particular threshold, we're going to see muscle cells commit to their pathways as muscle cells. And by using these linearized gene systems, we can figure out, well, where exactly is that threshold? Similarly, for immune response, we can figure out what is the threshold for antibody response, for primary immune response. And finally, for nervous system, this is very useful in investigating the phenomenon of long-term long-term potentiation, where we may want to figure out, what is the electrical potential at which AMPA receptors are going to be inserted into the neuronal cell membrane. So in addition, this is a major breakthrough for individualized gene therapy. So we may want to determine, the fine threshold, when does the drug take effect on cells, as discussed earlier. We may also we're finally able to actually fine-tune toxic drugs at viable levels, which is very, very important. Some limitations, miRNA does significantly decrease fold change and fold induction, so this is going to limit its applicability to systems that have high noise because we're not going to be able to see that difference between, maximal expression and no expression at all. Also, miRNA does appear to increase noise in some systems, which you can see on the right here. And the parameters for the miRNA systems were not experimentally determined or confirmed because all of this research was, of course, done from my home. All right, and finally, our future research aims. So in the research here, we used miRNA to degrade the mRNA, meaning that we're going to essentially block translation by affecting transcription. But in the future, we're looking to try blocking translation without affecting transcription. And we want to try doing that using a CRISPR-Cas9 system or CRISPR-Cas13D. And you can see here, the catalytic sites are removed in order to allow for that binding without affecting transcription. And we're hoping that this will preserve the fold change while lowering basal mRNA levels. So that was my project. Thank you so much for listening.